Really is a great privilege, and we have been trying for a couple of years to get her here. She is Canadian, originally from Waterloo, is it? Yeah, so maybe that helped to get her here. But she originally did her PhD at, her bachelor's in math at Ottawa, and then her PhD also in math at Berkeley. And then she joined the math department at Columbia as a postdoc, and then was hired by the computer science department at Columbia. And it was there at Columbia where she actually started getting into computational biology in the early 2000s. And now she is in the computational biology group at the Memorial Sloan Kettering Cancer Center. So Christina, come on up.
Okay, thanks so much for the invitation, and my apologies for being hard to book. It's just it's family travel constraints. It's not, holding out for large sums of money. Certainly not. All right. So it's great to be here, if only briefly, in Toronto, in the land of my birth. My lab is just to give a sort of broad introduction. We study regulation of gene expression at the transcriptional, co- and post-transcriptional levels, and the dysregulation of gene expression programs in cancer. I'm really only going to talk about one aspect of my lab's work today, which is the sort of cell-type specific regulatory programs, transcriptional programs, and epigenetic programs in differentiation. We also have activities in cancer systems biology and in microRNA-mediated regulation, alternative cleavage and polyadenylation, et cetera. All right. And I'm also not, I'm from a cancer center, a very exciting one, and I'm not going to give you a cancer program, but a story today, but just to motivate some of this work, what I am going to tell you about is decoding epigenetic programs in hematopoietic development and in particular in immune cells, in differentiation programs in immune cells. And this is very relevant in cancer. For example, quite famously in acute myeloid leukemia, there are aberrant developmental programs where you have a leukemia stem cell compartment that gives rise to sort of this altered developmental program, and there's a lot of interesting epigenetics to study here, and also immune cells in tumors. So this is actually a project we have cooking, I'm not going to tell you about, but even in very early tumors, T lymphocytes that infiltrate the tumor differentiate into a dysfunctional state, and that's why they can't, they aren't working, they aren't attacking the tumor cells anymore, and they present a challenge to immunotherapy. All right, so just with these kind of broad applications in mind, I'm going to tell you a little preamble story, which is how we got into the business of looking at epigenetic programs in differentiation. And for us, epigenomics means a bunch of different assays. The assay that drives our analysis or the assays are assays that map chromatin accessibility. All right, so in particular, DNA-seq and ATAC-seq. All right, so I think probably this audience is well aware of this, but just so we're on the same page, there are regions in the genome that are more accessible because transcription factors and other DNA binding proteins, structural proteins, are holding them open, holding the chromatin open. So, for example, this region that's maybe an enhancer or a promoter here is more accessible to an enzyme like DNase-1. And we can map the cleavage sites, or we can, in ATAC-seq, use TN5 transposase to sort of, in a tagmentation strategy, to get reads out of these open regions. And this, with a single assay, gives you genome-wide data on where are these accessible loci. And for us, it allows us to quantify, so by looking at reads at this event in different cell types, genome-wide, we can quantify changes in accessibility between two cell types. Okay? All right, so just a short preamble. This is published some time ago. We got into this business by looking at regulatory T cells. So regulatory T cells are very important, specialized CD4 T cells. They actually suppress inflammation, suppress the immune response. They're very necessary. If you don't have regulatory T cells, you would get terrible autoimmune disorders. They are unique in expressing this particular lineage-defining transcription factor FOXP3. Okay, so we have a specialized member of the T cell, CD4 T cell lineage, regulatory T cells, and they express FOXP3. And we wanted to study, well, if we look at precursor cells, okay, so you can think of just naive CD4 T cells as precursors, and they have to differentiate to regulatory T cells. At some point, they express FOXP3, and we wanted to understand how is the enhancer landscape changing in this differentiation step, okay? And we actually did this with DNase. This was actually part of Mouse and Code, and the data came from John Stam. And we were surprised when we mapped all the data, and we found this result. Okay, so here, every point is a DNase accessible region, a region of open chromatin. And it's quantified, we're looking at log read counts in, so this is FOXP3 positive, the regulatory T cells versus the precursor T cells. And you see there are some regions that are more accessible in regulatory T cells, there are some regions that are less accessible, but by and large, the enhancer landscape isn't changing that much, okay? By contrast, if you look at, say, CD4 T cells versus B cells, where there's a fork in the lineage, there's a massive changeover in the enhancer landscape, all right? But maybe this isn't so surprising. These are closely related cell types, but more surprising is that if you overlay these events with ChIP-seq for FOXP3, so remember, FOXP3 is only expressed in regulatory T cells, all right? But it's binding sites that were already accessible in the precursor cell type, all right? So we have a picture where, maybe before we thought, in order to get from one cell type to another, you express some transcription factor or cocktail of transcription factors that open up the chromatin and change the landscape. And here we see a sort of more nuanced picture where a transcription factor exploits a pre-existing landscape. And you have to do graphical abstracts for these kinds of papers, right? So sometimes there's another fork hit transcription factor as a placeholder, and then FOXP3 comes in, this one, or a cofactor is holding it open, or there's some changes through T-cell, TCR activation before FOXP3 expression. All right, but more broadly, we wanted to understand, is this exploitation, these early enhancer, is that a general principle of differentiation? And how does the organization and the dynamics of the enhancer landscape shape expression programs? All right, so with that sort of complicated preamble, what we did was we looked at human now, hematopoietic differentiation, and we just exploited free data, all right? So we took data from the Roadmap Epigenomics Consortium, and the data is technically of high quality, but it's not everything that you would want, all right? So if you're an immunologist or you study leukemia or hematopoiesis, it's actually a bit disappointing, but it's data that allows us to develop some computational machinery for this kind of problem. So what we have is human ES cells. We have hematopoietic stem and progenitor cells. These are CD34 cells. We have 3 kinds of lymphoid cells, and we have one kind of myeloid cells. We have CD4 T monocytes, okay? And we're missing a lot of steps in between, all right? So just as a warning, I'm going to talk about cell state transitions, and these are actually massive transitions in terms of developmental time. But since we just have precursor and then we have fully differentiated NK cells, we're just going to be talking about these steps. All right, so what I want to tell you about is first just some general principles of the spatial organization and dynamics of the enhancer landscape. And I'm going to talk about two ideas. One is regulatory locus complexity, and the other is early enhancer establishment that I gave you a peek at just now. I'm going to show you how to extract or predict transcription factor binding signals from DNase data. So if all you have in your cell type is chromatin accessibility, but you don't know what's binding, how can you computationally from the DNA sequence predict what might be binding, okay? And then we want to go from the enhancer landscape together with histone data, together with these predicted transcription factor binding signals, we want to be able to predict gene expression changes. Okay, so the last step is predicting gene expression changes in cell state transitions from the DNA content and lineage history of enhancers. Okay, and I told Lonnie I'm going to race because I want to show you unpublished work at the end of the talk. So, quickly. Okay. What we did was we took all these 6 cell types. We had replicates. We looked for events that were reproducible in at least one cell type. All right, and we created an atlas of DNase accessible events or DNase hypersensitive sites in the hematopoietic lineage to the extent that we sampled it. And we got an atlas of about 120,000 peaks or events. Okay. If you're going to talk about gene regulation, you have to assign enhancers to genes, okay? And we don't really know how to do this. So what we did was we assigned enhancers to the nearest gene. And we can discuss this in the questions. So that's problematic, right? So enhancers don't always regulate the nearest gene. But what is true is that if you just take genic regions, all right, and fatten them by 2 kb on either side, 58% of the atlas are in these genic regions. If you fatten by 10 kb on either side, 68% of the atlas is in these regions, okay? There are a lot of intronic enhancers. Half of non-promoter peaks are intronic, okay? So a lot of regulation is actually not so far away from genes. And then there are some enhancers that are very important that might be 2 megabases away, and that's true, but that is the exception. Okay, so once you do this, and you start looking at enhancers that are assigned to genes, you notice that there are genes like NUP54 on the left-hand side, which is a housekeeping gene, okay, nucleoporin gene. And you have other genes like RUNX1, which is a developmentally important transcription factor, okay? So NUP54 has only one peak assigned to it from the atlas, all right? So only one peak is near it. It's a promoter peak. It's constitutively open and active, okay? So this is a gene that just needs to be on all the time, and it doesn't need any fancy regulation to achieve that. Whereas RUNX1 has 43 DNase peaks assigned to it, okay? Many intronic peaks. I'm only showing you the first 4 exons here, okay? So the gene is actually longer. And you can see, I'm not telling you what all the colors mean. The colors are basically different histone marks, but you have lots of dynamics of accessibility and the histone marks across these 6 cell types, all right? So more generally, we want to capture this idea of this is what we would call high complexity, high regulatory complexity, and low regulatory complexity for NUP54. And just to define very simply this notion of regulatory complexity, we just take the number of DNase peaks across the lineage assigned to the gene, and we divide genes into 3 groups, high, medium, and low. And if you do that into 3 equal-sized groups of genes, it turns out that high complexity genes have 8 peaks or more, low complexity have 0, 1, or 2, and medium is in between. Okay? And these differences that you saw between NUP54 and RUNX are in general true. Okay? So if you look at the length of the gene, maybe not surprising, high complexity genes are longer. So this is on a log scale than medium complexity genes, which are longer than low complexity genes. Why are they longer? It's because more of the locus is intronic. Okay, high complexity genes have longer introns than and longer fraction of the locus is intronic than medium and then low. Okay. All right, so maybe this just follows from the definitions that we're assigning peaks to the nearest gene, but more is true, okay? So if you look at the dynamic range of expression across the 6 cell types, high complexity genes in red have the greatest dynamic range. So this is a CDF of that set of genes, and followed by medium complexity genes, and low complexity genes have the least dynamic range. Or if you look in a specific transition, all right, so hematopoietic stem and progenitor cells differentiating to monocytes, the genes that turn up the most are the high complexity genes, followed by medium, followed by low. Okay? I think I didn't say it, but it was on the slide. Highly expressed, high complexity genes are the ones that carry cell type specific information. So they're the ones that, the highly expressed high complexity genes in B cells have B cell annotations. They're enriched for transcription factors and signal transduction genes, whereas low complexity genes are housekeeping, translation, ubiquitination, sort of things that always have to happen. All right, so we have different structure, function, dynamic range of expression, and distribution of expression changes in cell type, okay, associated with these complexity classes. Okay, just one more point. You could imagine one of the things that goes wrong when you assign peaks to the nearest gene is that if you're in a gene-dense region, you don't get a chance to get many peaks assigned to you, okay? And we looked at density, okay, so you can just take the human genome and chop it into 1 megabase segments, and there's an enrichment of segments that are gene poor, all right? And this is a bit of a complex graph, but the basic idea is the high complexity genes live in gene poor regions. And the other part is that in gene dense regions, the peaks that you see tend to be constitutive. They're always accessible across all the cell types. They tend to be promoter or ubiquitous accessible peaks. Okay, so this is, as you get more gene dense, more and more of the peaks that you see, about 61% of the peaks in gene dense regions that are present in one differentiated cell type are present in all of them. Okay, so you have some kind of dichotomy in the genome, or gene dense regions that have, maybe there's a complex network of interactions, but it's constitutive. All these peaks are always there. And then in gene poor regions, you have these genes with very complex, many, many peaks regulating them, and lots of dynamics. Okay, so that was gene complexity. Now I want to get to the dynamics and to this notion of early enhancer establishment. Okay, so now let's look at the dynamics of accessibility of enhancers across cell types. Okay, and I'm just sampling from the 48,000 DNase hypersensitive sites that are present in monocytes. Okay, so they're all present in monocytes. And you can see some patterns that we know about, we expect. Okay, so promoter peaks tend to be ubiquitously accessible. Okay, always accessible. There are other ubiquitous peaks that aren't at promoters. Okay, there are cell type specific peaks. Actually, monocytes have a lot of them. Okay, so accessible in monocytes and not in any other cell type. But the thing that we were looking for and found, all right, are peaks that are present in the precursor in hematopoietic stem and progenitor cells, maintained in the cell state transition to monocytes, and lost in all other cell fates. Okay? So this is like this early enhancer establishment we saw with regulatory T cells. Something is set up maybe before it needs to be used, maintained in that transition, still accessible, but it's lost in the other cell types. Okay? All right. If you look at high complexity genes, genes with lots of DNase peaks across the enhancer versus medium versus low, and genes with just 0, 1, or 2 peaks, not surprisingly, low complexity genes are enriched for promoter and ubiquitous peaks. High complexity genes are enriched for cell type specific peaks, but also these transition peaks are enriched in high complexity genes. So it's not just that high complexity genes gain a lot of peaks in a particular cell type. Often they have this sort of transition enhancer. Okay? And here's another way of looking at that. Here, I'm looking at the transition from hematopoietic stem and progenitor cells to monocytes. Okay? Big step, a big differentiation step. And I'm looking at the high complexity genes that go up the most and go down the most. And we call these diamond plots. Okay, so the y-axis is the expression change, the log fold change, but then we put a stack of diamonds where every diamond represents a DNase peak. Red are peaks that are gained, blue are peaks that are lost. Okay, so part of turning up expression in this cell state transition is you gain a lot of peaks. Turning off a gene, you have to lose a lot of peaks. But you also see there are these yellow peaks and blue diamonds, yellow diamonds. These are transition enhancers that either gain or lose active histone marks. Okay? So the rule seems to be that to turn up a gene, a high complexity gene, you have to gain a lot of enhancers in the new cell type, but often there is this early enhancer, this transition enhancer that accompanies that change. Okay, so where are we so far with the story? We believe that complex locus control and early enhancer establishment are important for achieving these cell type specific expression programs. And in particular, this sort of transition pattern present in a precursor, maintained in one cell fate, lost in the other cell fates, is something we think is interesting. Okay? All right, so far this is statistics and genomics and properties, and now I want to do some modeling and a little bit of machine learning. Okay? So I want to be able to take all this epigenomic data and predict gene expression changes from DNA sequence of enhancers and also their lineage history. Okay? All right, so the first step is, as I said, we have free data, very high quality DNase data, but we don't have ChIP-seq data for any of the transcription factors that we might be interested in any of these cell types. All right? So where we don't have measurements, we have to predict. And what we do is, so we developed this method called seq-GL. GL is for group lasso, as you'll see. And the idea, actually, if you know my group's work, we've been doing these Kamer models for biological sequences and machine learning and classification problems for a decade or more. And we are using Kamer representations, so K-length subsequences of peaks as our positive examples and flanking regions as our negative examples. And we want to learn a model that discriminates between accessible regions, the summits, and the flanks that are inaccessible. And we want to do it in a way that reveals, many different transcription factors. Okay, so we want to be able to tease out what are the different DNA signals. All right, to get that idea of interpretability and getting different DNA signals, we do something actually quite simple. We take this, so here are our positive and negative examples, and here are k-mers, and we have a hit matrix, and we just cluster, okay? And what you see is you get this block structure, all right? So k-mers that are similar to each other or overlapping might represent different versions of a transcription factor binding site. Those k-mers will co-occur across examples. And we can use that to define groups of k-mers, and we put them into a group lasso, and then we use binary logistic regression to discriminate peaks from flanks. And what that gives us is a weighting over k-mers where a group is kind of all in or all out. If a group of k-mers doesn't help the discrimination problem, it's set uniformly to 0. Other groups are tend to be positive, associated with the positive class, and that's what we're interested in. Okay, so we have a weighted collection of k-mers giving us a signal for each of these groups, and then we can identify the hits, and then we cheat, we give it to Homer, because we need a name of a transcription. We need a motif, so we can look it up in a database and get the name, but we don't use this motif for any computation. The real signal, is a weighted collection of k-mers like this, and that's what we use for computation. But we still have this step where we need a motif, and we're looking forward to eliminating this step, but right now we use it. Okay, so the important thing is we can go from a DNA signal to a bunch of predicted signals of transcription factor binding. Okay, so this locus, we think PU1 binds here and here, CTCF binds here, so on. Okay, and we can see that it's making sense because, say, CTCF, the scores that we're computing are enriched in promoter and ubiquitous sites. Makes sense. Say PU1 is in transition peaks and cell type specific peaks in B cells. Okay. All right, so now the actual model. So we can go from DNase to a bunch of predicted transcription factor binding signals. How do we predict gene expression changes? Okay, so we're going to do the transition from hematopoietic stemmed progenitor cells to B cells as the example. Okay, so here's our starting cell type. We differentiate to here. For every gene, we have a set of peaks. Okay, and we also have predicted transcription factor binding signals. Okay, the interesting thing here is you could imagine maybe we have 50 transcription factor binding signals, and we just add them up across all the peaks, and we get 50 covariates for our regression model where we're trying to predict gene expression change, log fold change. We don't quite do that because transcription factor binding signals that occur in different kind of peaks. So if you're in a promoter peak, maybe you play a different role than if you're in a cell type specific peak or if you're in a transition peak. If you're a signal that is a placeholder keeping something open for later, that's different, okay? So we actually look at the DNA signals that occur in different kinds of peaks and treat them as different covariates. Okay, we have these 5 different kinds of peaks, and so we have 5 times 50 covariates instead of 50 covariates. That makes sense. Okay, but from there on in, it's rich regression. Okay, we also have second order terms. It's not so important, but this is just now a regression problem. We have different kinds of peaks. We have DNA transcription factor binding signals, and we're using these covariates to predict gene expression. Okay, how well do we do? Actually, you can do pretty well. All right, so especially for highly complex genes, this is a Spearman rank correlation on held out genes between the actual fold changes and the predicted fold changes, right? So 0.74, not bad, right? But what's more important than being able to predict the expression changes accurately is to interpret, what are the transcription factors that are orchestrating the expression changes? What does it all mean? Okay, so what I'm showing you here, I'm taking the input examples, so genes by transcription factor binding signals in different kinds of peaks. So you can think of this as more or less the input matrix, but then I'm weighting it by the regression coefficients that I learn. Okay, so orange says the model thinks these signals and this kind of peak and these genes are associated with upregulation. Blue means these signals are associated with turning the gene off as you differentiate to B cells. Okay? And then you see, this is interesting. What is this? So we have some B cell specific signals here, like IRF. We have some hematopoietic stem cell transcription factors here. Makes sense. But here we have these signals PU1, okay, in transition peaks. Okay, and if you look at these genes that have both PU1 signals in transition peaks and B cell specific signals, okay, so these are kind of like poised genes that have a transition peak that has a particular transcription factor PU1 that can bind. These are the ones that achieve the highest upregulation, stronger enrichment for B cell functional annotations, okay, and that data is here. Okay, so the poised genes turn on the most and they have the highest enrichment. So the model is telling us something about how genes are regulated and giving us a distinguished class of genes in B cell differentiation. Okay, good. One more point about the model. All right, so no one likes the idea that we map enhancers to the nearest gene because maybe it's not the nearest gene, maybe it's the second nearest gene or further away, okay? So if you actually look at the performance of the model, so you look at held out genes and you look at the actual log fold change versus the predicted fold change, okay? So pretty good, pretty good correlation, but there are some fold changes that were undershooting on either side, okay? So there's more upregulation than we can predict or more downregulation than we can account for. And when we looked to see what was happening, often it was a gene in a more gene-dense region with lots of peaks around it, but those peaks were being assigned to neighbors that were actually transcriptionally silent. Okay, we don't know why the neighbors are transcriptionally silent, but if you reassign those peaks to the right gene, then you can predict expression. All right? So you can just iteratively mask neighboring genes look for mistakes in the model, look for where our regression errors are high, see if there are neighboring genes that are transcriptionally silent, take those enhancers, reassign them, retrain the model. Okay, and that improves. But that improves performance. But now, you've already used your test data in that cell type to do this reassignment. So how do you show statistically that you're doing something good? Well, what you can do is take those rewirings, take those reassignments, and go to a different cell type, right? So look at T cell differentiation and use the B cell reassignments, and you can see that in this whole other cell type, you actually do reduce squared error. Okay, and just here's an example, right? So here's CD20 is kind of a classical B cell gene. It has these other genes nearby, but none of them are expressed. Okay, sorry, this is the RNA-seq. Only this gene is expressed, not the other genes. But there's lots of peaks all around. Okay, so once you start reassigning peaks, CD20 starts to have many. It becomes a high complexity gene, and then you can predict its upregulation in B cells. Okay, wrapping up this, and I'm happy that maybe it didn't make any sense, but I'm going to show you some new data, new project just now. But just to wrap up, okay, what do I think are the, how do we think about all this analysis and these results and this roadmap paper? So we think, well, how do multipotent cells encode the potential for different cell fates? We think part of it is this transition enhancer story, this early enhancer establishment. Okay, so you have these enhancers that are established in precursor cells and associated with a particular fate lost in other fates. And maybe that's for nucleating chromatin remodeling that is necessary before you gain all these cell type specific peaks. So we would love to test that with experimental collaborators, this idea of, nucleating chromatin remodeling. How do certain genes achieve highly cell type specific expression patterns? We think locus complexity is important. So if you want to turn up a gene really high in a certain cell type, you have to gain lots, you have to have high regulatory complexity. You have to have lots of enhancers. How can we accurately decode transcription factor binding signals from DNA-seq alone or ataxi? We use 6GL. We're working on refinements of this method, but we can, what I think is nice is that we're learning adaptively from the data. We're not taking a big database of transcription factor motifs that are redundant or imprecise or low content and scoring and getting a lot of false positives. Okay, so I think going from the data is better. Can we predict gene expression changes in cell state transitions from enhancer dynamics and sequence signals? Actually, it works surprisingly well, even though it's quite a naive model. Okay, and then this enhancer rewiring is learning from your mistakes to try to reassign enhancers to genes. Okay, so something unpublished, okay? We're actually taking all this machinery with the advent of ATAC-seq, which, in my earlier slide I didn't mention, but you have this new assay where you can get similar data of chromatin accessibility, but you only need 50,000 cells, whereas with DNA-seq you needed 20 million cells. All right, so now you have an assay that is feasible in rare cell types and clinical samples, and it really is empowering, and we are going to see things in the next few years that we've never been able to measure before. So some of the projects, what I'm going to tell you about is the first project, which is the furthest along. It's in review, the epigenetics of memory of inflammation in regulatory T cells. Okay, we're also working on projects like a tumor immunology project with Andrea Schietinger. Next year, maybe I'll be talking about this one. It's very exciting, the sort of dysfunctional state, understanding the dysfunctional state that CD8 lymphocytes adopt in tumors, even in very early tumors. We're looking at what's called an MLL rearranged leukemia. So this is an aggressive kind of acute myeloid leukemia and the role of a transcription factor called Helios. And we've just started looking in patient samples of doing ATAC-seq in pancreatic cancer to try to see if there are epigenetic subtypes of pancreatic cancer. Okay, so in the 15 minutes that I have left, let me tell you something new and unpublished. Okay, so right. So the notion of memory in the immune system is, fundamental and well established in certain classes of cells. Okay, so you could think you have some kind of immune challenge like an infection, and then followed by inflammation, and certain, CD8 cytotoxic T cells and CD4 T cells expand. Many they proliferate and fight the infection and cause inflammation, and then they many of them die, and then some differentiate to memory cells. Okay, so the notion of memory, CD4 cells, memory, CD8 cells is well established. So what about regulatory T cells, these kind of immunosuppressive cells that are important for preventing autoimmunity or autoimmune disorders? Do they have memory? Okay, and instead of what I'm going to show you in the next few slides, instead of looking at a typical immune challenge where we have an infection model, I'm just going to look at the response to massive inflammation, all right? And we actually will do that by transiently depleting the regulatory T cell population. So you kind of kill off most of the regulatory T cells, and you get massive inflammation, and then the population recovers, okay? And with very clever mouse genetics, so this is a collaborative project with Sasha Rudensky, who's the chair of immunology at Sloan Kettering, and always has beautiful data that we are privileged to analyze. So it's quite clever. So what you can do is you transfer 90% of these regulatory T cells that express a diphtheria toxin receptor under the FOXP3 promoter. So if you give diphtheria to the mice, those cells die, okay? But then 10% of the cells that you transfer are regulatory T cells that don't express the diphtheria toxin receptor, and they're labeled with YFP, okay? So you can do fate mapping. You can see these yellow cells. They were there from the start. So by, if you give diphtheria, you kill off most of the regulatory T cells. You get a massive inflammation by day 11, and these regulatory T cells are activated. They're actively trying to suppress inflammation. So they're in a different state. And then the yellow cells, 2 months later, they've recovered from the inflammation. Do they remember the inflammation? Do they change as a result of experiencing inflammation? Okay, and so the 3 cell types are resting regulatory T cells, okay, activated regulatory T cells, and this possible compartment memory regulatory T cells. Okay, so R Treg, A Treg, M Treg. Yeah? All right. Do they remember anything at the level of chromatin accessibility or gene expression changes? Okay, they do remember something, but it's actually quite subtle. Okay, so we have ATAC-seq data, and I'm showing you a meta-peak view. Okay, so I'm taking 400 peaks that are actually stably remembered. Okay, so they, are not there and resting. They become accessible in activated regulatory T cells, and they're remembered 2 months later. They're still there. But you also have changes that are transient and that are quite dramatic. Okay, so you have these sites that become much more accessible, but 2 months later they're back to where they were. Okay, so these are stable up, these are transient up, and you also have stable down, transient down. Okay, so some changes in accessibility are remembered, others are forgotten. Okay, what about gene expression changes? Okay, so again, so this plot, I'm looking at log fold changes resting to activated and resting to memory, all right? And if you parse it, you can see actually most of the expression changes are forgotten. Okay, so you have lots of expression changes associated, upregulation and downregulation associated with activation, and those are not maintained. Okay, the ones that are maintained are, it's a small set of genes that remember their activation and retain it. And if you look at what those genes are doing, they're actually associated with homing to tissues. Okay, so, and this is more clever mouse work where you can adoptively, you can co-transfer activated regulatory T cells or memory regulatory T cells with resting and T effector cells into another mouse, a lymphopenic mouse, and you can see where the cells go, and they go to these tissues. They don't go to the, relative to the spleen, they go to small intestine, liver, lung, okay? So maybe what they remember is where to go, okay, how to home to tissues, but they're not actually remembering their immunosuppressive capacity, okay? This is probably not even dark enough to see, but I just want to make the point that through sort of straightforward statistical analyses, you can see that the peaks and the histone marks that are associated with stable expression changes, so if you take the ATAC-seq peaks that are nearest to these genes that are stable up, they also become more accessible, and that greater accessibility is remembered, okay? And the histone marks are also remembered. However, the genes that are transiently, they're associated with activation and then they go back. The ataxic peaks associated to those genes are not remembered. Okay, so it makes sense, right, to some extent. So ataxic peaks associated genes with stable expression changes maintain their accessibility and their active or repressive chromatin marks, and ataxic peaks associated transient expression changes are not remembered. Okay? All right. So the point of memory in the immune system is that, you have a challenge, and then when you see the challenge again, you should be able to, the memory cells should be able to respond more efficiently, right? And usually we think of it as specific to an antigen. This is just a general inflammation, right? And something is happening in these memory regulatory T cells that, okay, so this is again a little bit complicated, but if we activate them again, if we take the memory cells and we put them in a new mouse and we reactivate them, okay, the genes that are just transiently up or transiently down, the first activation and the second activation look the same. All right, so subtly different reds are first activation and second activation genes that go up. Subtly different blues are genes that go down, first activation and second activation, and the lines are on top of each other. However, the stable expression changes are amplified in the second activation. Okay, so you can see, first activation, second activation. First activation, the genes that go down. Second activation, they go down even more. Okay, so there is something, there is some component of memory, but remember these stable expression changes are just a small part of the expression changes that happen when you activate regulatory T cells. Okay, so I think the story is something like this. Usually you think you have an immune challenge. We don't actually, what I showed you is not a classical infection model, but rather just we have this inflammation model. There's a transient increase in suppressive capability, but it's lost. And what's maintained is an altered tissue preference. So there is memory in regulatory T cells, but it's fairly limited. They transiently become more suppressive, and they have stable changes in tissue preference. And when you think about it, maybe you don't want stable changes in immunosuppressive capacity, or you would overreact. You would always be suppressing the immune response. But maybe you want to send these cells to the right places where they can respond to inflammation quickly. Okay, so lots of immunology. I'm not an immunologist, but some of my best friends are immunologists, and I enjoy working with sort of this amazing data on these very interesting questions. So just to wrap up, for the first part of the talk, I should have highlighted a few more people. So Manu Sethi and Alvaro Gonzalez were members of my lab who did the initial paper that I showed you. Manu's now in Donna Perez's lab at Columbia. Alvaro joined Will Greenleaf's startup called Epigenetic. He's at Stanford. And then Alvaro, as well as Hyunwoo Cho, did all the analysis for the memory of inflammation. And our collaborators, Sasha Rudensky and Joris van der Viken, an immensely talented graduate student in the Rudensky lab, did all the experiments. Okay, thanks so much. I'm happy to take questions. Okay, so the question was, I mentioned the Rick Young papers on super enhancers that actually motivated us quite a bit, and how is it related? So I think our definitions are a little different. I think Rick Young was onto something, is onto something. He defines super enhancers in slightly different ways in different papers, either like a big mediator signal or clusters of binding sites of master regulators for a particular cell type. He defines it as a cell type specific phenomenon. You know, in the right, you get the right thing to measure in the right cell type, and then there are these super enhancers. So we think of it more as the regulatory complexity. You look across the whole lineage, or ideally we would look across all cell types, and it's a property of the gene. So in a particular cell type, you might not see lots of enhancers, but later on you see those enhancers. So it's related. We were just, you know, perhaps just trying to simplify and codify, you know, just have a simple definition and see where we go with that. Yeah, but it's, we read all those papers while we were doing this work, and it's clearly important, I think, though it's also a rediscovery of a notion called locus control regions that, you know, have been defined before. So that's why we try to call it locus regulatory complexity, to, you know, refer to that older language. Yes.
So you mentioned ATOC can use 50,000 cells, and now there's single cell ATOC.
Yes.
Are you looking at single cell data, and can you comment on how that might relate to this memory effect? Is it, you know, is it a subset of cells? Is it, you know, is it a mix? Are we going to see a lot of differences between single cells, or do you think they're acting more like a bigger population?
Right. So we are thinking in those lines, right? So is that the subtleness of the memory cell, that it's associated with the subpopulation, right? We haven't looked at single cell ATAC-seq, and we're not actually planning to do it yet. I think it's very cool. Your dynamic range is 0, 1, or 2, right, at any site, and really just 0, 1, right? So you have this kind of noisy binary matrix, and you get power by summing over events, right? And I think the single cell RNA-seq is a little bit more mature, and we are, through actually Donna Pear, our collaborators are getting single cell RNA-seq, not in this system, more in a kind of tumor immunology, immune cells infiltrating tumors and seeing the diversity of cell types. And I think that's important, right? Just to see what is the ecosystem of immune cells that are in tumors. So, so I guess yes and no, not single cell ATAC-seq, though I think it's very cool, but certainly single cell RNA-seq.
So when you see particular transcription factors that are important for differential expression during cell differentiation based on your regression method, what do you think is the underlying mechanism? Is it that these transcription factors are differentially expressed, or we have differential accessibility maps for their binding sites, and the binding sites, for example, are blocked in one cell line?
Right. So I think it's a mix, right? So obviously there are some transcription factors that can create the chromatin accessibility changes, right? So that can remodel chromatin. And there are others that need partners to do that for them, and then they just exploit the changes in accessibility. So I think it's both. You can pose the question in a different way. So what we were doing was trying to take the enhancer landscape changes as the input and decipher the transcription factors that were orchestrating the change. But you could also ask, and we're trying to do this in various contexts, what are the transcription factors that are responsible for changing, you know, predict the chromatin changes, right? And I think one of the challenges is that it might not be that you're just trying to explain accessibility versus not accessibility in different cell types. Maybe it's important where you start and where you go to, right? So from one landscape you can get to another, right? But you get to the eventual cell type going through steps, right? And that history is important. So I think it's formulating the learning problem in the right way, and we're still playing with ideas.
So maybe another related question that I asked candidates: do you see transcription factor binding sites within constitutive DHS regions that still are informative about differential gene expression?
Okay, so you know, I would say we haven't explored the question fully, but in the monocyte differentiation, there's this CBP alpha, so there's a transcription factor that tends to bind promoters instead of enhancers. And so you see that signal. So it's not a transition enhancer exactly. It's not an enhancer that's just, you know, it's a promoter, not an enhancer associated with one cell type and lost. But yes, you know, the right transcription factor in the right context can, even via promoters, maybe with other partners, affect these changes. Yes.
So you said that the Tregs have a limited sort of memory. Was that a reference to your functional test, or how does it differ in type from the memory you get in B and T cells?
Okay, so I didn't show you any data because it's just, it's not published, right? Like we're able to look at these, the epigenetic changes or these, you know, DNA accessibility or DNA accessibility changes in activation in CD4 and CD8, and through various collaborators, Andrea Schiedinger and Sasha Rudensky, we've analyzed that data. It's not published. CD8 T cells naive to effector is the most massive enhancer landscape change we've ever seen in any cell type. It's like completely different cell type. We were amazed, right? And CD4, it's stronger than what we're seeing in regulatory T cells. So we're analyzing it. You know, we have the cell types. We have a lot of data that we have to plow through. But yes, and the other thing is that this general inflammation is not, you know, it's not the standard model. Usually you have an infection model, and it could be that if you look at antigen-specific regulatory T cells, and, you know, for infection models, we look at CD4 and CD8, right? And those changes are more dramatic, right? So, yeah.
We can take one more question, please.
So your chemo model is very powerful in the sense that you don't have to depend on the known set of motifs and things like that. But what I was wondering is, when you assign the label based on what PWS are out there,
Right.
are there important chemos you find that you have not been able to assign labels?
Okay. So
There are two TFs that don't know the secret.
okay, so in these fairly well-studied cell types, often is the other problem that there are multiple hits, right, that are similar motifs, different transcription factors, and we're not sure which one, or maybe, you know, maybe we haven't clustered properly. Maybe we need the whole thing to be more adapted, right, that we can relearn the groups because we're not getting a single clear signal. Other people have, like, so Anshul Kundaje used a version of SeqGL before it was published, and was looking in C. elegans, and he, you know, adapted the code, and he said they were finding novel motifs. So I think there's plenty to discover, in particular in less well-studied cell types. Though I would say, you know, it's a challenge. It's a challenge. You need more experiment. Once you think something is binding that you don't know what it is, then you have to figure out how to go after what it is, and that is some subtle experiments. Oh, thanks.
So Christina, we want to give you something in case when you're going back to the US you have trouble getting across the border. You can show this certificate that shows you were at an ISCB meeting and that you were a keynote speaker.
Do I need a passport if they have this?
Diane, do you know? But more importantly, here is a gold bar worth $10,000. So thank you.